This time we did a little bit of a different format. So we're doing this in a webinar format. So welcome everyone. I just want to let everyone know. So we have two expert panelists with me today. So Mary Gutierrez, who's with us last time, who's a virologist and has done a lot of work in hemorrhagic related fevers and worked in the BSL-3 lab and has a lot of experience and has done a lot of work with emerging pathogens globally to prevent just this sort of thing. So thanks for coming, Mary, who's in the East Coast, so she's a little bit later than us. And then Bernardita Mendez, who is a scientist and has really done amazing work along with her husband. And she's a PhD in cellular and molecular biology and did her thesis at UCSF and a postdoc in Berkeley and went on to co-found Chiron and has done a lot of beautiful work in this area and now is doing a lot of work in Chile, which she can speak more to, where she's built a research institute in an area to help researchers and entrepreneurs become more successful in their scientific work. So thank you guys both for volunteering. I appreciate it. So what I wanted to do is give sort of an update from last week to what's happened in a week. I mean, it's amazing what's going on. I feel like I think all of us that have been tracking this is our mouths in a fire hose of information, and literally things are changing day by day. I've just, just to update you, in addition to the clinical work, I've been doing and sort of my involvement with the whole PPE movement, where we created this online exchange, which we're about to launch, and I'll share with you guys later this week when it's launched, where we've been moving now N95 respirators from China and other places to hospitals here now in New York, Washington, and California. Which has been great. And the price points thankfully are coming down and things are starting to loosen up. And then the other part is I just got off a phone call with some scientists at UC Berkeley, because they're doing a seroprevalence study, and we'll talk more about this later on the slides, but they're interested in launching a seroprevalence study doing nasal swabs, self-guided nasal swab and finger stick, as well as from which they can get a lot of genomic material and do a random sampling of the Bay Area and do it sequentially over time. So we can actually start to look at acute infection rates, immunity, herd immunity, different genomic and features that make people more or less sick, and et cetera. So a lot of this work's going on. Stanford's going to do one, but they're planning to do it actually at a Home Depot parking lot. There's another university that's trying to do it at Walmart, so which we think is not, the UC Berkeley group is like, that is not we don't want people to be going out and being exposed. We want people to shelter in like they're supposed to. So their plan is to send kits to their people's homes where they can do it themselves and then package it up and send it back. Anyway, there's great work happening. There's huge cross collaboration, which is great. So, everyone can see my slides? I just want Mary and Bernardita, you guys can see this, yes?
Yeah.
Great.
Yes.
So I just want to show that it's doubling every week, right? So this was now 4 weeks ago. This was 2 weeks. This was last week, right? So a fourfold almost increase. And then from last week to this week, it's a doubling, right? If you just look at the number of cases, we have 900, almost 860,000. That was yesterday. And unfortunately, guess who's moved to the top of the list? Us in the US. So locally, this is what's been going on. And actually, this is available in the through the San Francisco Chronicle as well. But you can see here, Santa Clara has really been hit hard. And there's some reasons. People aren't totally clear what the reasons are, but there's been, there's a large Asian population there. There's been a lot of traffic movement through there. I think there was there's some other theories, but no one's clear, but clearly they've been hit the hardest. They're have the most number of deaths in all of California. Thankfully, you can see the East Bay, Marin County, and San Francisco is thankfully doing a lot better for now. And it looks like, we'll talk more about this. I think I have a slide on it. If not, I'll loop back to it, but it looks like we're doing pretty well. The latest update from think it's George Rutherford, who is at UCSF, who does a lot of this work and is involved with a lot of the modeling, is shown that right now there's 58 patients currently in San Francisco hospitals, and all the hospitals in San Francisco, which isn't so bad, right? Look at New York compared to us. And the trend looks like the trend's flattening for us. So that curve, we're sort of coming off that exponential increase in San Francisco and in the Bay Area. Santa Clara is a little bit of an anomaly, but the sheltering in, they think, is actually having an effect. It's far too early to release the sheltering in, but they're seeing an effect. And thankfully we did it, thanks to London Breed and others, a lot earlier than most. The other thing that people are doing a lot of research on is figuring out, you have the virus and then we have the host, us. And each one of us is a little different. You know, our microbiome's different, our set of immune system, how our immune system functions is different, our pre-existing health conditions, which we have found relates to how sick we may get, we don't know why yet, it will affect the severity of our illness. And that's all being researched across the globe right now in a very aggressive fashion. But it's really important to keep in mind this component. And, you know, it's a good reminder for me, myself. Last week, as many of you know, I've been really pushing hard, and I've mindfully said, like, okay, it's time to make a shift here. Speaking of which, what I'd like to do is spend just a few moments thinking of shifting and just do another meditation practice to build some resiliency. Resiliency is going to be a huge component for us. What I've realized, and we all need to recognize, is that this is not a sprint. This is going to be a several month journey, 3-6 months, and we're going to see what happens in the fall. And so it's time for a lot of us that feel like we've been called to arms here to really respond and do our best to contribute to really managing this pandemic as best we can, to really find a way to self-regulate and do this for the long run. So with that in mind, I'd like to sit for a moment and do a brief meditation like we did last time. So if you would, just uncross your legs and get yourself comfortable. And then just have a seat there. You can let your eyes be closed or open, whatever feels more comfortable. And take a big breath in and let it out. Let the busyness of our day settle out a little bit. Just to take a moment to notice your breath. And as your mind wanders, which mine just started doing, which it always likes to do, just bring yourself back to your breath, and I invite you to count your breath. Not so much each breath at a time, but within a breath, you can breathe in and count to 3 or 4. And exhale for a count of 4 or 5, so that your exhale is a little bit longer than your inhalation. One, 2, 3, 4, 5. Breathe in. 2, 3. And exhale. Just allow your mind, if it wanders, to come on back. And if you have neighborhood children wandering around like I do, allow it to float into the background. if you can, I'd like you to just take a moment to appreciate your current health, appreciate that you have good health and that we're able to stay healthy. And if some of us, which some of us have been sick, that we're mending our way back towards health again. And the importance of nurturing ourselves and getting rest, taking naps to allow ourselves to heal, to allow ourselves to build the resilience that we need to stay healthy. And if there's someone in your mind that you love, that loves you, just bring them into your mind's eye and let them fill you with that love. Let it bring a smile to your face, warm your heart. And if you feel capable, we can collectively take this nourishing, generous energy and love and put it out into the world to help those that are suffering now, that are sick, that are caring for the sick. Those that are our leadership locally at the state level and the national level, having compassion and providing some wisdom to those to help us find our way. And when you're ready, you can wiggle your fingers and your toes, bring yourselves back to our virtual room here. The beautiful thing about like this slide is really such a reminder for us that we can change our epigenetics. We can change our how our body responds to challenges. It's been really well documented that meditation practice and these resiliency practices, maybe it's yoga, maybe it's martial arts, whatever it might be, how they really actually can shift our neural response, our immune response, our stress response, and can help our healing and actually reduce the likelihood that we do become ill or the severity of our illness. So I encourage you to do a little bit of this every day. I'm glad we can do it together once a week for a little bit of time. And I encourage you to think about doing it. The best times of day often are in the morning upon awakening and just before bed to help you settle in and really provide a deeper sleep because the mind settles down a bit. So I'll keep moving forward here now that our host defense is up and we're feeling good. So the MMWR put out by the CDC came out recently, and they showed some really interesting findings, which is that we knew this from some of the China study, but we confirmed it here in the US too, which is diabetes, lung disease, and people with cardiovascular disease, which was loosely defined, not clearly defined, are at increased risk. So if, you know, what they say here is in the middle, basically, based on preliminary data, those with underlying health conditions like diabetes, lung disease, cardiovascular disease appear to be at increased risk. So those people should take particular caution. And you can see here the daily number reported of cases, right? This is reported cases that are coming through the CDC, going quite quickly. And then you can see, looking at the data, the main thing, I guess, when my circles moved when I projected this, but what we're seeing here basically is there are 74,000 cases. This database was based on only 7,000 of them, so 10%. And the reason is they couldn't find that no one reported the underlying health issues for the 90%. So it's a bit of a skewed database because maybe people are less sick and so they didn't report, or they're more healthy, and so they didn't report those underlying health issues. We don't know. But what's clear within this data set is that if you look at diabetes, for example, non-hospitalized versus hospitalized, so you can see here that there's an increased risk of being hospitalized if you have diabetes. And if you look at the bottom here, none of the above conditions. So 73% were not hospitalized if they didn't have any of these conditions. Whereas a larger proportion, like one or more conditions, you can see 70% were hospitalized. So if you just look at the numbers here, you can see just under the hospitalized, non-ICU. So 70% of those hospitalized had one or more health conditions, compared to only 30% of those that had no health conditions were hospitalized. So clearly having health issues is a risk factor, which doesn't surprise us, generally speaking, because people with health issues tend to be sicker and tend to have more problems in general. But this is pretty profound. And then they looked at it by age, and of course, as people get older, the risks are higher, and that's been pretty clear as well. One of the interesting findings, which I'll show you a little bit later, points to the fact that it looks like people as we age, we have higher viral titers at a given moment in time, like upon the first day of symptoms, for example. They're higher titers as you get older. So there's more viral burden on us. So what about the whole transmissibility piece? And Mary, I'm going to walk through some of this, and you may have some comments as I go through this. So I've been doing a lot of more reading about this. And so we know for sure that you need to wash your hands a lot, and we need to use hand sanitizer or soap. It's been amazing watching kids wash their hands. They're doing a lot more, but when you really watch them wash their hands, they don't do the best job. So they really require supervision. Those that have grandchildren or children that are young, or even for that matter, teenagers, we really need to show them how to wash their hands. They sort of go like that, and then they think they're done. Or they do this for, 2 minutes, literally, like, where they sort of just use part of their hands and they think they're done. So, and the other part is countertop. So that's not new information. Some of the part that's worth having conversations about, because this information is moving back and forth a lot. As you go to the grocery store, what are you supposed to do? At this point, my suggestion, which has gone against what I've suggested before, is people should be wearing gloves, and people should be wearing a mask. And I'll show you some of the data for why I think this is true. And this is going to go back and forth. And the national guidelines are changing, and part of it, if you look at the underlying reasons why they didn't suggest people wear masks, is because there was a low supply on masks, and they want to prioritize those as healthcare workers, which is understandable because they do have the highest risk. And now that the supply chain is changing, you'll hear Fauci and others start to have a different tone. And if you start to look at some of the data, you can see why this is so. And we'll talk about that in a couple more slides. So here's an example. This is UCSF on the left. It's the CEO, I believe that's Mark Larrett, and Josh Adler, who's the chief medical officer. They're wearing masks, and the reason they have surgical masks, which are different than N95 masks, which you can see on the right, surgical masks, they got a big shipment, 500,000 of these as a gift donated, and now everyone that walks in the UCSF is given one of these, whether you're a visitor or an employee. And these masks are helpful, primarily helpful by reducing the droplets that you may have if you cough or sneeze or when you're speaking, going out into the atmosphere. They're less helpful if someone coughs in your face, or you walk into, they're helpful. They're better than nothing, but an N95 mask is far superior to that. So certainly surgical masks are helpful. No one's done the head-to-head comparison between that and N95 masks, but we know N95 is better. I wouldn't suggest we hoard them, but I would suggest that we go out in the public, if we're going into grocery stores, not necessarily just walking down the street, but from close contact with people, we might want to consider wearing a mask. And the reason is, You can see here that, and that when someone coughs, droplets go out into space and it can be aerosolized. And it turns out they can hang around for quite some time. So initially at point 0, someone coughs, you get this big hit, if you're 6 feet, you're really close to them. And then you go to next time point, it gets more dispersed, right? And person B is inhaling these particles, person C doesn't have any exposure. And then over time, it's more dispersed and everyone's inhaling it. So the question is how far. It's a little bit unclear how far that is. But what is clear is that there's a difference. And this study showed, and this was published in 2017, looking at influenza. So it's a different virus. But what they showed is if you take people wearing N95s all the time, these are healthcare workers, or they just wear them when they're in the room with the patient doing something, or they're just wearing a surgical mask, which is what they put medical. You can see the difference in the likelihood of them becoming infected, or to say it differently, how well they're protected. So the bar that's going vertical that says 1 means no difference between two groups. As you move to the smaller number, you're getting protected. So 0.5 means you're a 50% reduction in getting, in this case, influenza. So you can see here for virus and influenza, you can see with the N95, it's further down, right? Down there like 0.5 or 0.25, much better than the surgical mask. And then the targeted N95 is a little bit better than always you wearing your N95 mask. So the other study that was done more recently looked at coronavirus 2, this virus, COVID-19, specifically. And this one showed that they did a test and they said, how long is it sitting in the aerosol, like in the atmosphere, aerosolized? And they did a 3-hour range and it was in there the entire time for 3 hours. So that says to me, oh, okay. So you cough, and Mary, you had brought this up privately with me. Someone coughs in one grocery aisle, you don't hear them cough or sneeze or whatever. And then 10 minutes later, you go into that grocery aisle. It's possible that it's aerosolized and sitting there suspended in air, and you walk in and breathe it in. The study also looked at different surfaces, copper, cardboard, stainless steel, and plastic. And you can see I put up top the guideline, the cutoffs there, 4 hours, copper seems to be the best, then cardboard, and then you have proceeding like that. So you can see plastic surfaces, it tends to linger the longest, which is why we want to disinfect stainless steel, plastic. When it comes to cardboard, there's a 24-hour mark. Now, what level of virus can you get infected? Like, is it sort of, once it's cut in half, which you can see, for example, with cardboard, by hour 8, it's cut in by, 10 a log of 3 down to a log of 1 by hour 8. How infectious is it? We don't know, but people did sweep up and find virus. So there's 2 main studies. The University of Nebraska did another study also and found that it was aerosolized as well. And again, the caveat that a lot of people are saying is we just don't know if that means how infectious it is. And also if someone's 10 days out and still shedding virus, they may have antibodies on their virus as it gets blown out into the atmosphere. And so that those antibodies make the virus basically dead, so it can't infect you. We don't know these answers. But in my mind, better safe than sorry. So those are my revised guidelines. And that's what I'm now recommending, which is, a big shift for me, and psychologically as well as in my recommendations.
Brad, if I could just jump in very briefly. Hello, everyone. I plan on conferring with Brad further on this because, as he said, there's lots of information that is coming out. It changes almost daily. I've been asked repeatedly about the use of masks, the use of respirators, and there are differences between these two, as I'm sure everyone is now fully aware of. We're all going to become experts at the differences between masks and respirators. What I will share with Brad is some pretty good information that I have on trying to delineate the various strengths of masks. If you don't have access to an N95, what we have now, very hot off the press, is information on the use of cloth masks. surgical masks, that some of those are considered cloth masks, versus these respirators. And this will help you determine whether or not you're using something that is deemed safe. One of the concerns with these masks versus, let's say, an N95 is how much do they serve as a barrier to these particles? And what you don't want to have happen is with these particles going into that cloth mask and now coming in close contact with your face and remaining there for a period of time while you have that mask on. So these are important points for consideration if you don't have access to an N95 respirator and you are thinking about a cloth, quote unquote, cloth mask. So I'll get with Brad on that, and I'm sure he'll be sharing that information with you as we compile it and as it evolves.
Thanks, Mary. So let me just pause for a moment. There's a few questions. So and if everyone can go through the Q&A rather than the chat, that'd be great. So a couple questions are, someone said that I've been running and biking and occasionally pass by people quickly, trying to stay 6 feet apart. That's pretty much my only exposure to non-family. I can't really wear a mask while running, although people are wearing masks while running, but yes. Would you advise against this activity? So I think if you're staying far apart, I think you're reducing your risk. I literally tend to hold my breath as I pass people, both for their benefit and mine. So sort of just pause breathing as you're going by. And I think that would be most helpful. The second question is, so I heard today about obesity as a group with increased risk, and you don't see that in the chart. That wouldn't surprise me, given that diabetes is considered a risk factor, and there's huge concordance between diabetes and obesity, although there are plenty of people that are type 2 diabetic that aren't obese, as well as, of course, type 1. But in general, that's true. So that really wouldn't surprise me. Also, a question about today's Nature paper, which I probably didn't see yet, shows that symptoms can include a runny nose. Have you heard this? I have heard that. And also up to 50% testing positive are asymptomatic. I haven't heard the statistics, I think, yet on, because we have poor data. No one has a seroprevalence. No one's doing seroprevalence studies yet. That's what I was talking about in the very beginning. Like, that's why I'm trying to help get this one study funded through Berkeley. If anyone's interested in helping get that funded, let me know, because we need this sort of data to know that. If you have a citation for that, please send it. Great. And then there's a couple chat questions. Where did they go? Can one use a N95 mask that you only use for a short time while shopping every day, or do you have to wipe it down and disinfect it? Do you want to handle that one, Mary?
Yeah, that's a good question. There are procedures that we are exploring now for reusing the N95s. Some of that requires a lot of work because you don't want to decompose the material that allows the respirator to function. We will share that information with you. What I do know they are suggesting in New York, for example, in the hospitals, is to have that N95 covered with a cloth mask, and then you discard the outer cloth mask, and that way you're retaining the N95. In terms of things that you can do to disinfect it, those are the things that I will share with Brad and he will share with you. As I said, some of it is quite rigorous and some of it is still up for discussion. So we'll try to get the best, most informative information to you regarding deconning.
Okay, great. So the other question is around N95s. Yeah, so let's see how this evolves and trying to figure out. I think as a good start, Mary, what do you think about surgical masks at a minimum?
Yeah, and I think if we abide by the table that I will share with you, Brad, which was put out by a mask manufacturer. And it will give people a sense of what masks they should be looking at. Because I will tell you, I went on Amazon. I've been receiving so many requests. I went on Amazon just to do a kind of an assessment as to what type of masks were being sold. And there's one mask on there that has the lowest proficiency, and they're sold out. And it concerns me that there are people out there thinking they're well protected with this one mask. So I agree with you. Surgical masks that meet at least the starting level of proficiency would be suitable, but anything less than that, no.
Great. And then
Brad,
yes.
could I interject something? For N95s, I'm assuming if we're looking at if people are using it to go out shopping or things like that, they would be masks that don't have a huge viral load. And at the same time, if we look at these papers that show that the virus lasts for, at the most 72 hours in plastic, and people are going shopping now, trying to go shopping once every 2 weeks or so, trying not to expose themselves that much. Would a possibility be just to let the mask sit out for 2 weeks and reuse it, not do anything? Because basically the virus is going to if there's anything there, the virus is going to disappear.
Yeah. So
I mean, that's a question. I'm just posing it.
Yeah. So the current practice for a lot of the hospitals, and I think it's University of Nebraska may have said this, who's written some of the protocols, is you get 4 masks, you number each one, 1, 2, 3, 4, and you hang them up. And you wear one mask one day,
Okay.
That's day 2. So by day 3 days, it's considered that safe, and so the 4th day is when you use it. So that's an easier method. That's and of course you're putting it on and taking it off from behind, right? You're not touching the mask. If you do touch a mask, it's with a glove, and then you discard the glove. Mary, were you going to say something?
I was just going to comment on that, because some of the procedures that are suggested are to do just that, to leave the mask alone, let it sit for a while, which works for the general public, as you said, Bernardita, those folks who are only putting on their mask every 2 weeks. Some of these other more intense, if you will, procedures are for those healthcare workers and others who are in the front line. So absolutely, you can apply some of those same principles that we do for the food that we're shopping for, the boxes we're bringing into our homes.
All right, let me go back, and I see there are more questions, but we'll get back to those in a little bit. So delivered, let me just actually, before we move on, so delivered packages and groceries. So Fauci said it's overkill. He was interviewed with Trevor, what's his name? Trevor, I'm blanking right now. And he said, Trevor Noah, thank you. And he said, look, it's overkill. You guys are, don't overthink it. But I would say in general, if you want to overthink it, which we're doing, you can have a little containment area, a little part of wherever you live, and just leave your boxes there for a day, and then go ahead and open them up. Or you open the stuff up, and then go wash your hands, and don't touch your face, or do it with gloves or something like that. Might be overkill, but again, I think better safe than sorry. And then another question was, why is the public not getting a mask, or not? Yeah, I think so. Clear, consistent, persistent sort of messaging about staying home. Well, I think California, they are, but I think unfortunately the rest of the country, it's a little bit more problematic, which honestly, as you can see, what's happening with China right now, although that's evolving too, I guess, but as soon as, if people are cross-fertilizing, we have a huge issue. So if I'm here and then I go to Florida and I come back here again, that's a problem. So in the duration of self-quarantine, once symptoms have subsided, in general, the thought, and we talked about this last week, there was that one study that showed people had viral load, that, it was decaying over time, but a third of the people still had virus by week 2 and well into week 3. So the current thought is 3 weeks. They were saying 2 weeks. The question is, like I said earlier, 2 weeks after symptoms have started, even though you have virus, the odds are pretty high that you have antibody on the virus. So it's probably not super active and infectious, but we don't know that for sure. All right, for the sake of time, I'm gonna keep going. So here's a presentation that some of the new things at the bottom here: loss of taste and smell, and up to one study showed a third of the people have a change of taste and smell. Some people reported, rhinorrhea, which is a runny nose, sore throat, headaches, GI symptoms. Some people have only had GI symptoms is how they started. And then later, and if you look here, fever, which was really the only way they were going to test us in the beginning, was a requirement of fever. Only half the people had fever on admission into the hospital. So that just shows you that there's a full range of symptoms that are possible. Which makes this a bit more alarming. So my take home on this is, don't overthink it. Don't drive yourself crazy. We're going to start noticing everything about ourselves and being really afraid. Do your best to just sort of roll with it and let those things sort of come and go. Take care of yourself, and then you'll notice when something all of a sudden shifts in a big way. Where you're all of a sudden feeling fatigued and ill, in which case, please reach out to me and Julia and all of us here at the practice. So David, I think, couldn't make it. He was going to text me if he could make it. He was going to tell his story because he's positive for it. So let me just tell David, if you are there, just send me a message or text me or something. But let me tell you his story because he said I could. He's basically started off, well, he went skiing with a group of 10 guys. 8 of the 10 guys got sick. He went helicopter skiing, so they were in small quarters together. One of them was in the hospital with COVID-19. The other one, at the time he contacted me, which is several weeks ago, he knew was positive as well, and several people were feeling ill, including him. And so we did the swab. It took 10 days to get it back, unfortunately. And the day prior took 5 days. So we actually swabbed people and the day prior. And then we ended up it coming back. And what happened to him for his story was big cough, sweats, some nights sweats, some nights not sweats. Big hacking cough was a predominant one. Had in the beginning a bit of a sore throat and a headache, which went away. Significant fatigue, and in particular, which I've heard this a lot, lying down, really significant coughing. So keeping up at night, in particular, a lot of coughing whenever lying flat. Now, people get that in general with a bad URI as well, but this is pretty profound for him. I think we both think he did have a bit of a pneumonia with this, not significant enough to need to be in the hospital or require the oxygen concentration machine, which he actually did have. And we did start Plaquenil and azithromycin, which I'll tell you more about later. Whether it's coincident or not, a day or two later, maybe 3 days later, he started feeling a lot better. And now he finished his last day of the medicine too, and he's feeling significantly better, but not yet completely healed. So that's 3 weeks in. So how long is someone infectious? So you can see here, days after onset of symptoms. So the question is, so can you grow it in a culture? So this is one study that looked at that and looks like, days after symptom onset, you can see here that the positive cultures started to wane after 8 days. So that leads us to believe, maybe people are only infectious for the first, call it week to 10 days. One study So there's a lot more data we need to confirm that. This was the comment I made earlier about age and how as we get older, the viral loads tend to go up, and maybe that's why people do less well. Well, let me reword it. It's a surrogate marker for why people aren't doing as well, right? Our immune either are, they may have the same initial burden that was put on them, but they are having a harder time fighting the virus, and so the virus is growing in their body. So it's a higher load. So, and then labs, which is less relevant, more relevant for me, but I just thought I'd throw it in there. The bottom line here is very little lab abnormalities. If anything, the main thing is people have a drop in their white count. That's the main thing that people notice. And so let's move to diagnostics. So there's two big sort of areas. One is get the virus and how much virus do you have in your body? And the other is, do you have antibodies? The virus one, again, we want to get it as soon as we can. For example, I tested myself, but it took so long to get the test, 17 days after my symptoms started, test was negative. I don't know if if it was too late or not. And so I can't wait to get the antibody test because I want to see if I have antibodies to it, right? I think a lot of us are going to be in that camp. And a lot of us just want to know, period. In general, how's our testing coming along? Well, this graph sums it up pretty easily. And this says that we, at the national level, really dropped the ball. And which honestly is why we're in our current predicament, because we totally dropped the ball. We decided that we were going to develop our own test rather than use the WHO test, as well as not heeding the plan for significant, intense contact tracing, test, contact trace, and isolate people. And so now so many people are positive that we're having a hard time catching up. So the direct detection of the virus is really for just the acute diagnosis and to determine if they're still infectious. And that's when we talked about last week, the nasal swab that goes to the back of your throat, or your pharynx. And there's locations that are around that are doing that, as we talked about last week. UCSF is really ramping up their volume, which is great. Marin General has it as well from people in Marin, and there's a few other centers as well. The other one is antibody detection, and I'm really excited about this. And you guys may have seen there's a couple companies now that have FDA clearance to be able to promote their work. It's still, honestly, there's still the sensitivity and specificity on those tests. They've only done a small number of people, so we don't quite know the accuracy of these tests. There's some preliminary data, so I thought it was pretty good enough that the FDA said, go out and get it, you can sell it. So that's where we are now. I've ordered tests already through Henry Schein, who's the distributor for the SD biosensor test. BodySphere just announced that they have a test as well. And I put an inquiry into them to see what the differences are really, and I haven't heard back. These guys, Henry Schein, are gonna get access to these SD biosensor tests by Monday, they said, and then we'll ship it out from there. I think they're getting 50,000 or 500,000, I forget now. So we should be getting some of those. Please, so I'm first going to prioritize those that have had symptoms and use the testing, but please do let us know if you want to be tested, if you've had symptoms of some sort. And then of course, if you haven't had symptoms, I'm happy to do it as well once we sort of gone through the top list of people that have had significant symptoms and want to know if they're immune. But please do let us know. You can email Donna, and then we can sort of move from there, and we'll keep a list of those that want to be tested. So this is what the machines look like. Cepheid is one that's down in the valley, it's Silicon Valley, that was FDA cleared. They're trying to get their test up and running. They have these big machines. The Abbott ID is another machine that UCSF is using. And they can do upward of 1,000 a day, is my understanding. And then there's the finger stick testing. This is for the virus. So this is where the swabs doing the PCR testing. The serology testing is a finger stick. This is the stuff this is the BD biosensor that we're going to use. And a little finger stick with a lancet, and then you drop the blood into this little well, add some reagent to it, and then you get your results. And look at IgM, which is sort of the first thing that comes online for our antibodies to fight something, then IgG, which is longer term immunity. We don't know. Does that mean you're immune for years? Does it immune for only for a little bit? That part's unclear. So the other, let me just move to surveillance data. So this is the call I just got off of with some of the faculty at UC Berkeley, is different people are looking to do different surveillance studies. Some people are repurposing blood banks that they have already that are population-based data to then see if they can identify, if it's recently pulled. For example, Stanford had a whole bunch of serum from people that they're coming in for influenza, checking for influenza. So they went back and saw what proportion of those people at the time had COVID. I think it was around 5%, if my memory is correct. So there's a lot of folks that are repurposing existing data. The study that I'm looking into and looking to help raise money for is going to actually randomly sample people right now in their homes, and which is basically a much better way to do it, to identify, get both genomic data as well as nucleic acid data, as well as for antibody testing as well. And then do that over time so we can see the shift in the population over time as we build herd immunity and start to get people back to work and back into school, et cetera. So again, we talked a bit about the tests. Unfortunately, Quest and LabCorp have huge delays. They're trying to fix that. That renders me being able to do the test much more difficult. So we've been making a lot more use of Stanford and UCSF in particular. So let's talk about some of the treatments. So there's a lot of excitement about these HIV drugs, this combination drug, which turns out to actually a recent randomized controlled study, that's what the RCT means, showed that there was no benefit in New England Journal of Medicine. It was published. So that's been put to rest. At least maybe someone else is doing the study too. We'll see if they have different outcomes, but a lot of people sort of put that aside as a result. You've all heard about the Plaquenil azithromycin. And again, a lot of us that are in the scientific community that understand science are saying, look, this is a study based on 30 people. Who knows? The guy lost 6 people to follow up. We talked about this last week. There's another study that came out of China that said it didn't work at all. And there's a guy that said he's treated 699 people, and he claims not a single one of them need to go to the hospital. But it wasn't a study. It was just him saying, like, this is a group of people I treated, and it worked. But there's lots of problems with that. So there are lots of studies going on. We'll talk about that right now as we speak for that combination of medicine. Then Remdesivir is a Gilead drug that's IV infused. There's an adaptive study going on right now. I'll just sort of forward to that so you can see that. It's an NIH study over multiple locations throughout the US. It's an adaptive study, meaning that as soon as they get findings, they're going to tweak the study. They're going to add new treatments into it. They're going to change the dosing of this existing treatment. Whatever they find, they'll quickly adapt and keep the study going, which is great. That particular study is hospitalized patients because it's IV within 72 hours of being hospitalized. They got to be relatively severe, which is usually why they're hospitalized anyway. It's a 10-day treatment, initial bolus, and then 100 milligrams daily. And those are some of the endpoints they're looking at. UCSF has only had a few patients, but they've rapidly enrolled. They're almost full already. They were looking to get 440 patients. And they're, I'm sorry, it's also in Singapore and South Korea. So they're almost full. And they're going to quickly then, as soon as they get data, switch and tweak the study based on their findings. And then the other one is the Plaquenil, the hydroxychloroquine. There's a dosing finding pilot study going on. Those are some UCSF investigators. And then there's a couple other studies that are looking at it, as well as this other study looking at high-dose vitamin C versus Tylenol for acute respiratory distress in the setting of people who have usually an intensive care unit for that, and they're intubated. And that's been going on at UCSF. And then just back to the other options, there's this convalescent serum idea, which they used in the olden days. I think it was with Spanish flu. I'm trying to remember where else they used it. But you take people that got sick, but hopefully very sick, actually, because they have a huge antibody response, and then got better. And then you take their blood and you process it and put it into people that are sick. And basically you're harvesting their antibodies and putting it in sick people. And there's one study of 5 people where they did this, showing in New England Journal of Medicine, showing benefit. And so there's a lot more interest in this. And so there's some studies going on with that. There's a range of other studies, as you can see here down the list. Of note, people were doing steroid treatments. That's been shown to actually worsen people's condition. So that's no longer something that people are offering, which just shows you it's really important to study this stuff and not just do it on theory, because you can make people worse. So I wanted to, I guess I'll pause for a moment and just see if any of the other panelists, is there anything you guys want to add? Or are you guys good? Okay.
Not at the moment, Brad. I'm fine.
Okay. So hold on, I need to. Pull this back up. I lost. Can you guys see okay now, the slides?
Yes.
Great. So just to go back to ibuprofen, there's a lot of conversation about this. Unfortunately, there's a lot of doctors claiming that the ibuprofen's bad. I think WHO came out and said it as well for a little while. There's no data to support that. It's theoretical. And so people that need ibuprofen should be taking it. With that said, it's possible that it does harm, but we don't know. There's also some in vitro studies showing that it actually may help people. So the data on this is out. I'm not a huge fan of NSAIDs in general anyway, because they increase cardiovascular risk anyway, and they increase risk of bleeding in the stomach, and they increase, kidney problems as well. But, people need it for short-term use, and so they should use it when they need to, and I wouldn't recommend that you not use it. If you need to use it daily, we should have that conversation why you're needing to use it daily. But there's just be advised that there is no data, even though doctors are claiming there's data, there is no data to support that claim. The other area of a lot of conversation is around ACE inhibitors and ARBs. These are a class of drugs that are used for the heart and for heart remodeling, and also for blood pressure control. Like, same thing, there's some theoretical risk around this, because we do know the virus goes into the ACE2 receptor. And so the thought is, if you're on these ACE inhibitors, you have more receptors, and so the virus has more doorways to go in through. But actually, there's also data saying the opposite, that actually, since there's a re-regulation of the receptors, because they get upregulated and then they get downregulated, that actually it may help people with this virus. So it's unclear right now, and all the national bodies are saying, don't stop your medication, don't change it. We just don't know. There is a theory that a couple people I've been talking to have said, like, if you are on this ACE inhibitor, think about it. You basically, what it does to you is it makes it so that it's harder for this ACE inhibitor to have something attached to it. And so then the body automatically adjusts and creates more receptors in response. Now, the medication still overwhelms those more receptors. But if you take someone off it, then all of a sudden you have a whole bunch of receptors, maybe 2 times more than you normally have in real life. And now if you get sick, and that theory is true, now you've got a real problem, right? So the same thinking about, for example, people that take PPIs, proton pump inhibitors for their stomach, and when you stop it, they get a worsening of their reflux. The same reasoning is because these receptors get upregulated. So right now, let's keep doing what we're doing. And then just for the sake of time, I'll just keep moving through here. I want to talk about vaccines, and I'd love to get Bernardita's view on this. She's done a lot of work in this area in the past, and understands the regulatory issues around it and a lot of the challenges that one has. I sort of listed one. There's a huge list. This website I put at the bottom here, bioworld.com, has a huge list of treatments that are emerging, therapeutics that are being looked at, and a huge list of different companies doing different vaccines. So why don't I pause for a moment, Bernardita, if you want to weigh in, I'd love to hear your views.
Okay, fine. So first of all, just to mention, as Brad said, that there are efforts, the whole listing of the 20th of March has of the order of 40 ongoing projects in vaccines. Of those, there are 2 that are already in clinical studies. And
questions while we wait for her to come back. Is childhood asthma a concern as it relates to vulnerability? Is one question. And the answer is yes. We don't know that. We know that in adults that's true. Kids in general do great. If you're asking if you once had childhood asthma and now you don't, I think the odds are low that it'd be a problem for you. But we don't know that for certain, but that would be my best estimate. Question, are you immune when you've had the virus? The thought is yes, at least in its current rendition. Again, we don't know, it's new for all of us. So that's what we want to see. But in general, that tends to be true. That people, once you get something, you get antibodies to it, you tend to do good. Now, if you take the flu as an example, though, you know, it mutates, and so then you're not immune for the next flu. Same with the common cold. But for this particular, right now, we don't think it's mutating a lot. And so odds are that if you have antibodies, right, the current plan is like you go back as a healthcare worker. For me, for example, I'm going back into the war zone and I'm taking care of patients again because I have immunity to it. From my workplace, do thermometers work, and do you have a recommendation? So I'm a little less clear about the use of thermometers. They were used widely in China and Korea, but in general, they do work, and people at a mass level, they're a crude tool that does work. If someone has a fever, they do not belong in the workplace. But remember, you're not going to get everyone, right? So half the people upon admission, half the people upon admission into the hospital didn't have a fever. But they did use that as a screening tool. So let's see what else we've got. We're all stuck at home, shelter in place. Our houses are getting stuffy. Will opening the windows help keep our homes healthier and keep our immune system stronger? Yeah, so
okay. I'm back, Brad. I don't know. Murphy's Law. Of course, it has to disconnect the minute I had started talking.
That's all right.
Hey, and Bernadette, turn your video off if we have bandwidth problems. That'll help.
Yeah, but it just happened just suddenly. It didn't announce itself at all. So anyway. So that should be fine now.
Yeah. So we lost you. You said there's a list of 42 in clinical trials.
Yeah. So basically 2 are in clinical trials and 1 should be starting up in April. So the interesting thing is, the question is why 42? What happens is basically to make a vaccine, you can use different technologies. And what's happening is, every single one of these, either companies or combinations, coalitions between public and private, NIH and NIAIDs, have expertise in a particular kind of technology. So basically, we are right now moving forward in parallel with as many possibilities in the different technologies as we can. So what is a vaccine? Basically, what we want with a vaccine is to expose a person to an attenuated form of the pathogen, so the person can build up an immune response. So historically, in the old times, the only way to do that was to use the pathogen and either kill the pathogen or attenuate the pathogen. So to date, we have many vaccines that are attenuated or inactivated viruses. And in fact, one of the technologies that we're testing or we're developing as a potential vaccine is an inactivated viral vaccine. That's one. But with the advent of molecular biology and recombinant DNA, the new ways of developing vaccines or developing new technologies is to do bits and pieces of these pathogens. Mainly the piece that the body sees, which is the surface protein, the coding of the virus. So you can do that by giving our bodies either the surface protein, the protein of the virus, or giving other pieces that allow your body to produce the viral protein. And that is why we have, for example, vaccines that are DNA vaccines, that if you put them in a person, eventually the person is going to produce a protein, a viral protein, and the body is going to mount a response. So in that sense, we have vaccines that are DNA vaccines. We have vaccines that are RNA vaccines, which is also genetic, but another sequence, another type of sequence that's going to do the same. It's going to allow our body to produce a viral protein, just a protein. It's no infectious at all. It's just a piece of the virus. And different kinds of technologies. So we're talking about like 6 different technologies. These, there are today, there is something that's called CEPI, that it's important because it's a coalition for epidemic preparation innovation that was formed in 2017 in Davos. And the purpose of this organization was particularly to develop vaccines for a safer world. We're a little late on that, but these guys are basically focusing on 8 different projects, and they're funding them pretty heftily. So, you know, we're talking about in the order of millions of dollars, 10, 9, 30, 40 million dollars. So from the standpoint of, I would say, feasibility of the initial project up to the clinical phase and including the clinical phase, this project should move along. Now, in terms of the projects that are in clinical phase, the question is how long would they take? The projects that are starting now have a time frame that goes all the way through March to June next year. And at that point, what we will know is 2 things. One is whether the vaccine is safe from the standpoint of whether it produces any type of adverse events. We will also know whether it produces local reactogenicity. And we will look at the antibodies that people are producing from 2 standpoints. In general, what type of antibodies it produces, to what, if we can detect the antibodies, and also whether the antibodies that are produced are blocking for the virus in vitro, because we can look whether the virus can infect cells in the lab, in a Petri dish, and we can look whether these antibodies have the property of neutralizing the virus. So that's the information that we'll have by the sometime first quarter next year or mid next year with these ones. It may be that the next round of studies that start up a little later, some of them can be a little faster, I don't know. But basically that's the time frame that we're facing. The 2 that are most advanced, one is the one that Brad showed, which is Moderna, which is an RNA vaccine. So you put a genetic sequence in the person and the person's system produces the viral protein that elicits the immune response. And that is a study that's going to be done in the US at different doses and so forth. The information is all available. I can send links if somebody's interested. And there's another study that's being done in China already with another kind of technology that's a viral vector technology that is similar in a way. It allows the body to produce a protein that's going to generate the immune reaction. So I would say that's kind of the general lay of the land. There's a fair amount of information that's available. I think that the 2 organizations that are focusing on this is CEPI, and it's also Gavi, which is the organization that's associated with the Gates Foundation, that are very much looking into the overall vaccine development.
Thanks, Bernardita. Can you talk a little bit more, help us understand the RNA vaccines? It's sort of a novel method, right? Are there other examples of that? And
there's no RNA vaccine approved in the market. So this would be a first. So basically what you do is it's like thinking in a concept of a pen drive where you put a program and you put it in a computer. The computer takes the program and basically is able to act on it and has a product. So what you do is you take this, you put it in the body reads the program, is able to produce a protein. This protein is the viral protein, that is the spike protein. It codes for the spike protein. The spike protein is the protein that basically attaches to the cell and allows the virus to enter. But if you have the spike protein, because it's in the surface, it should be very immunogenic. So what would happen then is once you have that in your body, you produce the protein, your body sees the protein, and then you produce antibodies against the protein. In that sense, then, once you get the virus, you are already prepared. Those antibodies block the virus. Those antibodies attack the virus.
great, thank you. Awesome.
I'll just add to that, Bernardita, that was wonderful, thank you. We are seeing these vaccines, candidate vaccines that are being tested right now for hemorrhagic fever viruses, which are what we call some of these RNA viruses. And I know I worked on some early generation or versions of these vaccines maybe over a decade ago. Mm-hmm. And we're really excited about them coming to the forefront. It's great body of science, so very cool.
Yeah.
Great. Thank you both. And the take-home message in my mind about all this is it's not going to solve our issue for this round, nor the fall, and we need to, hunker down.
It's definitely going to be after that. So we have to put our bets on antivirals or monoclonal antibodies.
Yes, I agree.
Yeah.
But some good news there. I think back to when we were managing, I don't even want to call it managing, when we were responding to the West Africa Ebola virus outbreak. They came up with a solution. The virus emerged in 1976, I think is the first published outbreak. And when the community got together in 2014, they came up with an antiviral so fast for something that we thought we could not get an antiviral for. And it happened. And right now, I will tell you, and I'm sure Bernardita and everyone else who's in the science community will tell you, I don't know a single scientist or a single clinical researcher who's working on anything else but Corona, SARS-CoV-2, COVID-19. Everybody in the world is working on it. So it's amazing. I think we will see those antivirals. I'm counting on it. Hopefully by the fall.
I also think that now, basically machine learning and artificial intelligence is so much better than when we were talking about the previous epidemics. So that part is being also very helpful in terms of doing pre-screening, in silico pre-screening of drugs. So yeah. I think we should be able to get something earlier than fall.
Yeah, I agree.
Thank you. So let me just, there's one more question I want to address, which was, we're all stuck in our homes. So, and what are your thoughts about improved ventilation? So that is a big issue. And I encourage people to open their windows, use proper, HEPA filters as well, and to really do your best to really allow for ventilation, as you know. And I think, Bill, if you're still on the call, there's someone called the Hayward score. Bill Hayward, who is one of the members here at Blue Wave, one of my patients, a great guy who has really devoted his life now to the quality of the home and what goes on when the quality of the home and how people get sick. And he tells a story about his own experience with that, with mold and what went on and sort of the trials and tribulation of that experience. And as a result, has really done a huge amount of work around this. So I encourage you to look at the Hayward score, to look at your own home and evaluate the quality of the home. And I would say that keeping your windows open and bringing fresh air in is really important. There are people that come into your house, your kids, the range of people that come through at some level. It's important to have some ventilation, let alone the side effects of being in the home and the mildew and the heat and all the stuff that happens along with that. That's due to the sort of the cleanliness or lack thereof of the home, not so much on the floors, but what's under the floors, what's in the basement, et cetera, things like that. For the sake of time, it's 5:15. I might spend a moment. Dave, thanks for joining, David, and we can see you. You're one of our panelists. Do you want to speak to sort of a bit of your story? There's a tutorial that we're going to do on the oxygen concentration machine that's happening now. I don't know if that's going to kick us off or just maybe hold people from being able to get onto this, which is fine. But David, why don't we take advantage of the moment and just share a bit of your story, since you're willing to volunteer and people I'm sure want to know.
Yep, happy to, Brad. Thanks. It's interesting because I've been hearing stories since then and they're all sounding very similar, but was on a trip with 9 friends to go skiing in Canada. So somewhere, on the way there, somebody in the group or someone on the plane probably had virus. No way to know. 8 out of 9 of us got symptoms pretty quickly, and 8 out of 9 have tested positive, 1 tested negative. And we all went through this pretty much exactly the same way, which was basically, all the symptoms of a very bad flu, fever, chills, body aches, the whole bit, and kind of went up and down. Some people slept, couldn't get out of bed. Some people could, and then that turned into a pretty bad cough. With all the hallmarks of pneumonia that was difficult to suppress. It did not produce anything, and it got worse at night and couldn't lay down. So my situation was I wasn't really able to sleep very well. So that went on, and I met with Dr. Brad to get tested, and I probably tested a week into it. The results took about 2 weeks to get. By that time, it was very clear that I was probably positive. And then I did, at the doctor's orders, although probably a week later than he suggested, took the hydroxychloroquine and the Z-Pak, and whether I was 14 days into it, which was the case, or and getting better, or that kick-started it, I immediately began to feel better. And I would say about the course that was prescribed the time was 6 days, of which 4 days include the antibiotic. About 5 days into it, markedly better. Each day was a little bit better, but 5 days into it felt completely better. And then, now I'm 3 weeks into the entire process, and pretty much symptom free, and according to the health department, cleared. So hope that's helpful.
Thanks, David. And I'm glad you're on the other side. What he didn't speak to is he couldn't even speak. He couldn't be on any calls. I mean, just speaking alone, he would start coughing and really, and for David to get David to slow down has been challenging. And so this finally, he had no choice.
Yeah, it felt like someone was sitting on your chest. So every breath was labored, and then speaking would trigger a cough, which would trigger a really quick spike in temperature, up to some serious, 104 type temperatures. So, at some point that became the straw that broke the camel's back.